Okay, this is after we got the sample prepared. We need in order to investigate using next generation sequencing, we need to generate the library. There are several steps. The first step, I didn't put in the schematic picture. The first step is try to digest those DNA fragments. That's using this DNS I to digest those DNA fragments. Then, after we remove all the DNA fragments, we can use this poly-T, this kind of grid to enrich those mRNA. As I have mentioned, the structure of mRNA, it has a long tail of this A nucleotide. So we can make use of this characteristic and using the braid, this T braid, this poly-T braid, to enrich all those mRNA, and then we will try to fragment the mRNA into pieces, and then we will use cDNA synthesis to get this double strand one, and then we will cut it. We will repair the end and put an A on each tail, and then do the ligation to adapter. And after that, we will do the PCR amplification. And after all these steps, this library is ready for sequencing. So here, this should be day 2. I'm sorry for this mistake. So this is how we did in the library generation. And after we got this library, there will be a quality check. And this is some standard of our quality check. And the sequencing strategy. Here, I'm mostly talking about this transcriptome, both the novel sequencing and resequencing. We will use such a sequencing strategy. That is, this P indicates the pair-end sequencing, and this 91 means we will sequencing 90 nucleotides from both ends. And I here just mentioned our another product, RNA level product called codification, which is using another sequencing strategy, which I will mention it in the last part of this presentation. Here, let's just skip this. And recommend data for each sample is 4 giga base pair or is 22 million pair and reads. So after we collect the sample, after we generate the library, then the next step we need to do is try to analyze all the very short reads. That read is only about 90 base pair long. So this is our pipeline of bioinformatics analysis. The first one is this is the read we got from sequencing machine from the Illumina HiSeq, and then we using this very short read, the first thing we will do is filtering. We will try to filtering those reads with very low sequencing quality, and also we will try to do alignment to the anti-database, try to figure out whether there's contamination. And if there's any contamination, we will filter it out in this step. And also, we will try to filter out the adapter sequence. And after the filtering step, we got the clean data, the clean read. And making use of this clean read, and we will do the de novo assembly and try to get the unigenes, or maybe also called transcripts. And got a side of unigenes, we will then use kind of alignment to the database and try to do the gene annotation. so as to know what's the functional role of those unigenes. So whether it is investigated or not. And also using this annotation information, and we can kind of calculate the expression level of each unigene, and we can know the function. We can calculate the expression level, and also we can use this function annotation to make gene prediction using this way. If we have more than one sample, that's probably we can compare the expression level of one gene in different samples and try to figure out whether there's differentially expressed gene. And for those and also for those differentially expressed gene, we can further figure out what's their function, whether it is related to the biological process we are concerned about or not. We can do the geo-enrichment analysis, and also we can do the pathway enrichment analysis. This is a general idea of all the process. I think I will give some detail about the technician. This feature, this slide I just mentioned is about de novo sequencing. You know that there is some de novo assembly of the RNA sequencing rate, and there is an annotation of all those genes in this transcriptome. And in this slide, this is the schematic picture of the resequencing pipeline. So here is the raw data. This is from our sequencing analyzer, and then we also have the filtering the low quality one, filtering the contamination, and filtering those adapter sequence. And after that, there's this sequencing. This de novo assembly step. We don't have to assemble it. Here, the resequencing one means here is a reference genome or maybe a reference transcriptome. So we just have to map all these clean data to the reference genome or reference transcriptome, and we can try to statistics the coverage, and we can try to get some information about how the distribution of this sequencing rate on the reference genome or the reference transcriptome. And also we can calculate the expression level of each unigene. We can kind of refine the gene structure, and we can detect alternative splicing, and we can detect whether there's some novel transcripts or the information of single nucleotide polymorphism using this resequencing alignment. And if there's more than one sample, we can try to compare the expression difference between different samples, whether there's one gene is downregulates or upregulates in different samples, and what's the function of those differentially expression genes. So this is a general idea of the whole pipeline, and I will give you some more information about the technician details. So this is something about how we do the de novo assembly. We know there's some challenges in this transcriptome de novo assembly because some characteristic of this transcriptome, because the transcriptome is uneven coverage. Some transcripts are highly expressed, thus we will sequence a large amount of RNA sequence, and some of transcripts are very didn't express that much, so we cannot sequence too many transcripts. So those transcripts have very low coverage. This uneven coverage causes a problem for our de novo assembly. And also, because this uneven coverage, the sequencing error becomes a huge problem, because the sequencing error in those high expressed region can be very abundant, even abundant than those correct sequence from the low coverage transcript. So this is the second problem. And also because we all know that there is alternative splicing in the transcriptome. So since most of assemble method is based on the Brio graph, this alternative splicing will make the Brio graph really complete, really hard for us to solve. And also, this repeat sequence, this is the same difficult using NGS technology to assemble the genome and as well as the transcriptome. This repeat sequence will make us very hard to decide whether this part should be is a repeat sequence. It should be assembled into one contig, or it should located in different compartments. Different contig. It's very hard to decide it. So this is several difficulties we face. And now here is the Trinity. I think most people might maybe heard about this software. This is widely accepted the novel assembly software, and the name Trinity means that's a combination of three compartments. One is called Introduction, and the second one is called chrysalis, and the other one is called butterfly. And the combination of these three compartments is just like the stage of how an inchworm turns into the butterfly. So this inchworm is trying to use overlap information and connect the very short sequencing rate into longer and longer contigs. So this is what they did in the first step in this inchworm software. And then this chrysalis will use the contig is already generated by this inchworm, and use this contig and try to build the Brio graph for each cluster of related contigs. Then this we can get this the Brio graph, and then the butterfly is try to resolve the Brio graph and try to figure out whether there's some different spliced isoforms and whether it's a paralog transcript. So this is schematic picture of the whole pipeline of this software Trinity. So this is the first one, is the inchworm. It is trying to disuse overlap information and connect very short sequencing rates into this longer linear contig. So each color represents each contig. So this is what they did by the first software called inchworm. And then the second one is trying to use the contig generated by this inchworm and try to use the contig information and cut it into a very smaller camera and try to use the similarity between different cameras and generate this the Brio figure, the Brio diagram. So after you got this information, can represent the complexity of this transcriptome. And after this chrysalis step, then use the butterfly, we can figure out which should be coded into two separate transcripts, and which part represents different alternative splice. So as we can see, here is one isoform, here is one transcript, and this is this one for this red, and then it's blue, and here also has this transcript also has another isoform. Here is a very short, kind of short blue one, and here is a longer blue one. So this can be assembled into two different isoforms. And since here the coverage is really low, that means this transcript should be cut into two pieces at this location. Well, this is another transcript, and from this Gabriel diagram we can see here is one exon, and here is probably another one, and here is another one. So we can get this if we connect all these really longer one, this is probably one isoform, and since here is another blue one, if we connect these two, here is another isoform, and also about this blue one. So this is how the Trinity work on the novel assembly of this transcriptome using the very short trees. That's using overlapping information, connecting to county, and try to use county to generate the Brill diagram, and try to solve the Brill diagram, try to figure out where is the transcript and which different isoforms are there, and got a set of transcripts for this species. This is a whole pipeline. And after we use this Trinity to de novo assemble the transcriptome, we can make use of this transcriptome and try to align all the transcripts into some known database, such as Interpro or the KEGG pathway, or some other known database to figure out what's their function. So this is here is some figure I try to show you the geo annotation of all the transcripts in one transcriptome, and how many genes located in one function and how many genes located in another function. This is what we can do after we've already got the novelly assembled transcriptome. We can do the gene prediction, and we can do the functional annotation. And another thing that is of very important, of a lot of importance in this in this RNA in this de novo transcriptome sequencing is try to figure out the expression level of one gene. Normally, in our project, we will use this RPKM or this FPKM to represent expression level. This RPKM represents rate per kilobase of exon per million map rate. And this FPKM have a similar meaning because it doesn't count the number of reads, it counts the number of fragments, which had even wide concept to this RPKM. So this is the formula used to calculate the RPKM value. This is the expression value of an unit gene A. So here is the total rate mapped to the exon of this unit gene A. So this unit gene A, and it should divide it into the mapped reads, all the mapped reads to the transcriptome. this is the all the mapped reads to the transcriptome, to that is to all the unit gene, and then also this number should be divided into the exon length. So this kind of formula kind of eliminates the influence of different gene lengths because the mapped read number is divided this gene length, and also eliminates the influence of the sequence level because this is the parameter, this is the value represents the sequencing data, the sequencing level, and this is the parameter represents the different gene length. So using the number of reads mapped to into one unit gene and divided the number of reads mapped to all the unit gene and divided the length of this unit gene can allow us to know the expression level. And this is kind of normalized expression value, and we can just use this expression value to compare in different samples. Okay, so after we using RPKM or FPKM to get the information about the expression value of one unit gene, something we even care about is whether this gene is upregulated or is downregulated in some biological process or in some special stage or in some special tissue. And we try to relate this upregulation or downregulation to some specific biological function. So this is why we're interested in this differentially expressed gene. So we have developed some in-house software and developed some really strict requirements to identify, to detect those differentially expressed genes. Here, normally we will use this false discovery rate and use this number, and also we would like to say there should be at least twofold expression value change. That means the expression value of this unit gene in sample A and in sample A and sample B, the RPKM value or FPKM value should be twofold difference in this, at least twofold difference in these two samples. Then we will think this is a differentially expressed gene, and we will choose screen it out as candidate gene. This is a scheme. This is a picture from an ongoing project. We calculate the number of upregulated. This is 3 samples, and in each sample we'll try to figure out how many genes are upregulated and how many genes are downregulated. And for those upregulated gene and downregulated gene, we need to further understand its function so as to understand what kind of physiological process or what kind of biological function is related with it. So this is we try to map those differentially expressed genes to the KEGG pathway. So the red color means this gene is upregulated, and this green color means this gene is kind of downregulated. And after we got this kind of KEGG pathway figure, we will know which part of this pathway is being shut down or maybe being activated in this pathway. and this can help us to answer some biological questions. And with this de novo sequencing, de novo assembled transcriptome, or with the resequencing mapped transcriptome, we can also use to refine the gene structure. I'm sorry, I just made a mistake. This gene structure refinement only used in this resequencing the novel resequencing a transcriptome, because you need to first have this reference gene model, and you need to have the reference genome. Then you use the resequencing read map to the genome and try to figure out whether the expressed fragment is corresponding to the exon in your reference genome. As we can see, this is almost correct, but for this one, and the reference genome, the exon as exon is only this kind of shot, but we can use resequencing, use transcriptome resequencing. We can see here are some recovered cover this region. That means this region is also expressed. That means this region should also be an exon. So the resequencing of transcriptome can be used to refinement the gene structure and try to extend the exon, the age of exon. So we can get better gene model using this method. And the other problem we can solve using the novel transcriptome or resequencing transcriptome is try to detect alternative splicing and try to detect whether there's some novel transcripts. So this is one of our ongoing projects, and here is some alternative splicing we detect, and we count the number of this kind of unigene and try to count the number of these alternative splicing events. So this IR means intron return. That means in some of the isoform, this intron is transcript is expressed, and in some kind of isoform, this is removed from the mRNA. So this kind of event compares 24% of all the alternative splicing events. And here is another kind of alternative splicing event that is some change in this 3' end. So some of the isoform is start the second exon start from this point, and in some isoform, the start point of the second exon start from here. So this is another kind of alternative splicing event we call ALT3' SS, and this is another one. It's kind of similar from this one, but it happens in the 5' end. And also some of these alternative splicing events is kind of coupled with each other. There's a combination of 2 or more splicing events, and we can kind of count the number of this situation and count its proportion. So here I use several cases to illustrate how we making use of this novel assembly of transcriptome to investigate some in some plant and animal projects. So this is the novel assembly of the transcriptome of peanuts, And here is the background information. We all know that peanut is very important oilseed crop, but the peanuts still don't have a reference genome because it's tetraploid and it's allotetraploid. So it's very hard for us to use NGS technology to generate a reference genome for this species. So if we the novel assembly of transcriptome will greatly improve our opportunity to understand this species. This is how we decided the investigation. They choose 3 different peanut varieties, which is similar, immature, precise, but their oil content is kind of different. For the GH4 variety, the oil content is 57, and this KO1 is 52%, and this T21 is 48%. So they try to use the de novo assembly of transcriptome and try to compare the transcriptome of these 3 peanut varieties, try to answer the question why there's some difference in this oil content. The sequencing strategy they use is P101, that means sequencing 100 nucleotides from both ends, and this P is parent, and using this Illumina HiSeq. And the novel assembly software they use of the novel, and as a result, they got this number of unigenes for the peanut transcriptome, and by compare the expression level of different unigenes, they detect several key genes that involved in fatty acid metabolism and biosynthesis. In this kind of precise, there are several genes. There's 146 genes that is related with this function. It shows differential expression level. So using the novel assembly transcriptome, they got a reference transcriptome for peanut, and also they reveal several genes that is very important in this fatty acid metabolism and biosynthesis precise. So this is a paper published in 2012. So this is for the novel assembly result. This is the name of different variety, and this is the total sequencing rate they generate for this project. And this is the number of base pair, number of nucleotides. This is how they statistic the disease content, and this is the number for each variety. So using these 3 varieties together, they got that by 59,000 unique genes. So this is their assembly result and what they got from this project. This is another project about the Newen. So the Newen is a very good model organism to study regeneration, because we all know about oh, I'm sorry, there's okay. I think there's some problem just now. Okay, I'll continue this presentation. So here is some background information. Some background information about this project. The Newt is very good model organism to study regeneration, and also because the genome size of this species is really large, and we still don't have a reference genome for this species, and that's why they decided this project to novelly assemble the transcriptome, and also they try to sequence the protein profiling and try to do some validation. The sample they use, since they are interested in the process of regeneration, the sample they use is undamaged one, and also from undamaged one, and also from the lung regeneration tissue. And they try to figure out what kind of genes is involved in this regeneration process. And this is how they collect the sample for the RNA project. And the sequencing for the sequencing platform, they're using NGS 454 platform, and they also use some protein samples and using this MS-based technology for proteomics. So as a result, they got this number of transcripts for the Newt. And but by using all these transcripts aligned to some known database, they can annotate this number of transcripts. And also, since they've already did the proteomic investigation, they use their proteomic results to kind of experimentally validate this data. They detect, and the most important part of this project is they detect some novel protein families and some novel proteins, and found these proteins are probably related with the regeneration ability. And the validation is by protein profiling, and they also find this expression difference using transcriptome, which use RNA-seq data. So what I just talking about is how we sequencing the mRNA. The messenger RNA, those RNA can be translated into protein and can be involved in some biological process. And I try to talking about how we collect sample and how we generate library and how we did sequencing and how we did bioinformatics analysis. The difference between these two, the novel transcriptome and the sequencing this transcriptome, is only in the last part in the bioinformatics analysis. The novel assembly transcriptome, you need to maybe use some software such as Trinity or maybe some other software to assemble the transcriptome. But the resequencing one, since you already have a reference genome or you already have a reference transcriptome, you don't have to do the novel assembly. You just have to map all the sequencing rates to the reference genome or transcriptome. So this is the only difference. And both the resequencing transcriptome and difference and the novel transcriptome can help us to figure out the expression value of some specific gene and try to help us figure out which gene is kind of upregulated or downregulated in different samples, maybe in different tissues or maybe in different stage. And also both the resequencing and the de novo sequencing of transcriptome can help us to detect alternative splicing. So that's all about the messenger RNA sequencing. In the second part of this presentation, I will try to talk about some non-coding RNA. Here, especially, I will talking about small RNA, which with a kind of shorter RNA length, and also the long non-coding RNA. All this non-coding RNA is really important in the biological process or in the development or in the or has a regulational role in translation or some post-translation. And also we will try to research try to tell something about sample test, about library construction, about the NGS sequencing, and also about the bioinformatics and analysis. So this is the small RNA sequencing. The small RNA sequencing, we care about some short RNA fragments that is its length ranges from 18 to 30 base pair to nucleotide. And in this study, we mostly focus on the microRNA or called microRNA, which is 22 in nucleotide, and it can be found in both plant and animals and have very important function role, such as in the translation transcriptional regulation and post-transcriptional regulation of expression. It can affect the degeneration of other messenger mRNA, thus to affect the expression value of some genes. So this is how we this is a sample collection and how we generate a library. So the sample condition that is required is total RNA, and so this is the quality of the sample, the concentration of the RNA, and also the library construction. We will try to get all RNA in length in this range. And sequencing strategy we use is IC50. That means single-end sequencing, only 15 nucleotides. And for each sample, we will try to sequence this 10 million rate. This is the pipeline of elaboration library construction. This is total RNA, and we try to firstly do the selection of size, and we try to use scale to get the RNA fragment length from reach from 16 to 28 nucleotides, and then we do the ligation to adapter at both 5' end and 3' end, and after we do the ligation to adapter, we will do the PCR amplification of this RNA fragment, and after the RNA amplification, we will do the sequencing. So this is the bioinformatics analysis pipeline for this small RNA sequencing. So this is the raw data. This is the data from sequencing analyzer. And so the first step is your filter. Try to filter the low quality rate. Try to filter the adapter, and try to filter those with contamination. And after all this filter, we got the clean data, and we do a quality check. If it satisfy our requirements, then those clean data, those single end sequencing RNA data will map into the genome. We will try to, using this alignment to the genome, we will try to figure out the distribution of RNA fragments along the genome, and to check whether and also to check whether the quality is satisfy our requirement is reasonable or not. And if everything is good, then we will try to align all those really short small RNA sequence to some database such as this MyRBase. This is a collection of microRNA, and also to the index database, and we try through this alignment to all the database, we will figure out which gene, which RNA is already known in those databases. We can detect all those known RNA, and also we can detect whether these RNA, whether these are this small RNA located in the repeat region, or in the exon region, or in the intron region. For those small RNA, we cannot annotate using the alignment to those databases. We will try to further investigate its secondary structure and try to detect some novel microRNA, and also try to predict, try to use their similarity to other region in reference genome, and try to detect the target its target gene. For those already known small RNA in our sequencing rate, we will try to do some other analysis, such as this whether this microRNA is added or not, which family it belongs to, and also we can calculate the expression value of this microRNA and compare with different samples, and try to figure out whether this microRNA is upregulated or downregulated in different samples. And also, we can do cluster analysis. That is, after we got the differential expression genes, we can cluster those genes with similar expression change, and those microRNA with similar expression change pattern is probably related to similar function, or maybe involved in the same biological process. That's what we did in this in the bioinformatics analysis in the small RNA sequencing. And here is a case for this small RNA sequencing that investigates all this barley. And some background information about barley. It's very important cereal crop, and the microRNA research of these species, there's not too much investigation about this microRNA of this species, and this is the design of this project. They got RNA from 4 different tissues, from the root, the stem, the leaves, and spikes, and they use those samples generate to construct libraries and use Illumina to sequencing it. And the sequencing strategy they use is a single end and sequencing about 15 nucleotides. And as a result, they got this amount of clean reads and they got this amount of unique sequence. And the most importantly, they identified about 126 conserved microRNA, and they belong to 58 families, and also and they classified several microRNA that is very important in the drought and drought stress response. So here is a schematic. Here is a picture of their results. They found this microRNA 0 to 9. That's this one. That is drought stress related because in the drought sample it is kind of changing expression level, and also they found this this 3 is 156 and 396 and this 399. It has it increases expression level and the drought stress, suggesting they are involved in this response to the drought. So this is what they did from the small RNA sequencing of barley. Here is another part. This is about non-coding RNA sequencing. What we just talking about small RNA sequencing, the small RNA has a line switch from 18 to 30 nucleotides, but this long non-coding RNA is normally has 200 nucleotide in length. And this long non-coding RNA also has very important functions. They are involved in various biological processes such as epigenetic regulation, and also they are involved in transcriptional and post-transcriptional regulation. And this long non-coding RNA sequencing allows us to sequence both long non-coding RNA and also mRNA. I will explain this later in the library construction you will see. And we can calculate, we can try to sequence the long non-coding RNA and also calculate its expression level in different samples and try to figure out whether the expression level is changed or not in different samples, in different states or in different tissues. So this is the workflow for long non-coding RNA sequencing. Here we can see this is the total DNA. This is the sample after we digest the DNA fragment and got the total RNA. And for this total RNA, we don't remove those mRNA in this library construction. We only remove the rRNA, ribosome RNA. That's because after this we can have the idea that we will got some mRNA and also we will got some long non-coding RNA. We didn't remove those messenger RNA because some of the non-coding RNA also has a poly A tail. So if we use the poly A tail to enrich those messenger RNA and remove it from the non-coding from the total RNA, that's probably we will lose several important long non-coding RNA. And that's why this long non-coding RNA allow us to sequence those messenger RNA and also the long non-coding RNA, because we only remove the ribosome RNA here. So after we remove our RNA, we got the RNA fragment, and we do the ligation, and we do the cDNA synthesis, and then here is the size selection. We will select the size that is about 200 base higher, because that's the length of the long non-coding RNA. And after this size selection, we will do the PCR amplification. And after all these steps, we will do the Illumina sequencing of all these RNA. And this is a bioinformatics pipeline for the long non-coding RNA sequencing. This kind of analyze, including some blood alignment to some known database, and try to identify some already known long non-coding RNA. And also, since we have the mRNA messenger RNA in the sequencing rate, we can use the messenger sequencing RNA to do the transcript assembly. And also, after we've already got the annotation of long non-coding RNA, we can calculate the expression level and try to compare the differential compare the expression level of long non-coding RNA in different samples. And also, according to the RNA secondary structure, we can detect whether which family this long non-coding RNA belongs to. And after all these steps, we still have some long non-coding RNA. We cannot get any information from all alignment to the known database or from the secondary structure. It's probably some novel long non-coding RNA. We can detect some novel one. This is a example of a long non-coding RNA sequencing of the zebrafish. Some background information about the zebrafish. That's very important model fishes. And this investigation mostly pay attention to the embryogenesis stage. They try to detect whether the long non-coding RNA expression RNA change their expression during this embryogenesis process. So this is their design of the experiment. They choose 8 different developing stages of wild zebrafish embryos. This is the 8 different stages, and they try to sequencing the long non-coding RNA in all these 8 samples, and they use this Illumina HiSeq 2000, and they use the strain specific library. And